Good day, everyone. Our topic for today is all about ketogenesis and ketone bodies. Ketogenesis is the biochemical process through which organisms produce ketone bodies through breakdown of fatty acids and ketogenic amino acids. This process supplies energy under circumstances such as fasting or caloric restriction to certain organs, particularly the brain, heart, and skeletal muscles. Ketogenesis, it means synthesis of ketone bodies. The site of this ketogenesis is mitochondrial matrix of liver cells. Reaction. The reaction of ketogenesis is the major pathway of ketone bodies for formation is HMG or hydroxyl methyl glutari CoA Lay's pathway. The reaction of this pathway are we have the 5 reaction. 1. Formation of acetoacetyl CoA. Second, we have the formation of beta hydromethyl glutaryl CoA. The third, formation of acetoacetate. Fourth, we have the formation of beta hydroxybutyrate. And the fifth, we have the formation of acetone. The formation of acetoacetyl CoA, we have the two molecules of acetyl CoA condensed to form acetoacetyl CoA. This reaction is catalyzed by thiolase, an enzyme involved in the final step of beta oxidation. Second, the formation of beta hydromethyl glutaryl CoA. In this reaction, acetoacetyl CoA combines with another molecule of acetyl CoA to produce hydroxyl beta-methyl glutari CoA, or what we call HMG CoA. The third reaction, formation of acetoacetate. HMG CoA lays cleaves HMG CoA to produce acetoacetate and acetyl CoA. Fourth, the formation of beta hydrobutyrate. In this reaction, acetoacetate can undergo spontaneous by a decarboxylation to form acetone. We have the last, formation of acetone. In this reaction, acetoacetate can be produced by a dehydrogenase to beta hydroxybutyrate. So, let's proceed to the regulation of ketogenesis. Regulation of ketogenesis is regulated mainly by, we have the 4 regulation. First, we have the substrate availability. The substrate availability increased ketogenesis occurs when there is excessive availability of fatty acids for oxidation. Thus, increased ketogenesis occurs during starvation or diabetes mellitus. Second, we have the regulation of beta oxidation. In this regulation, it increased glucagon and decreased insulin in fasting result in inhibition of acetyl CoA carboxylase. This result in decrease in malonyl CoA. Decreased malonyl CoA results in increased beta oxidation of fatty acids or activation of carnitine palmitoyl transferase. Third, we have the availability of ATP. It increased beta oxidation results in more production of ATP through citric acid cycle in liver. In this result, in increased availability of acetyl CoA for ketogenesis. We have the 4th regulation of ketogenesis. 4th, introduction of HMG CoA synthase. In this 4th regulation of ketogenesis, HMG CoA synthase is the rate-limiting enzyme in ketogenesis. The synthesis of HMG CoA synthase is stimulated by fasting increased intake of fat, and diabetes mellitus. Fatty acids are strong inducers of HMG-CoA synthesis. The increase of synthesis of enzyme occurs by increased transcription. So we have the outline of ketogenesis. First, two molecules of acetyl-CoA are condensed to form acetoacetyl-CoA by acetoacetyl-CoA synthase enzyme. To that one, more molecules of acetyl-CoA is added to form HMG-CoA by HMG-CoA synthase enzyme. Second, we have the ketogenesis and their utilization is taking place even in normal conditions, but both processes occur at slow level in such a way that their normal level remains in 1.0 milligram. Third, we have the ketogenesis starts from mitochondrial acetyl-CoA in liver. Synthesis is mainly hepatic in nature, but liver, due to absence of enzyme thioprase or acetoacetyl-CoA transferase, is unable to utilize Kb as source of energy.
Yo.
Ketogenesis or formation of ketone bodies is an alternative catabolic pathway for active acetates. The amount of ketone bodies is small in normal individuals, but their levels become important in certain metabolic conditions. Acetoacetate and 3-hydroxybutyrate and acetone are all ketone bodies. In acetoacetate, the structure is CH3CO CH2COOH, while the beta 3-hydroxybutyrate, we have the CH3 CHOH CH2COOH, and the acetone, we have the CH3 COCH3. The next, we have ketone bodies. Ketone bodies are metabolic products that are produced in exist during excessive breakdown of fatty acids. It is water soluble and produces energy yielding. In a normal man, the concentration of ketone bodies in the blood is usually less than 3 mg to 100 mm. Significance of ketone bodies. The significance of ketone bodies, we have three. The first significance is alternate source to glucose for energy needs. In this, ketone bodies represent an alternative to glucose for the provision of energy to the cells. We have the second significance of ketone bodies. Production of ketone bodies under condition of cellular energy depletion. In this significance, the ketone bodies are produced when excessive fatty acids are being oxidized. It is because of increased adipose tissues lipolysis, and glucose availability to the cells is limited. The third, utilization of ketone bodies by the brain. In this significance of ketone bodies, the brain normally utilizes glucose for energy needs. Although ketone bodies cannot completely replace glucose for energy needs in the brain gets adapted to oxidize ketone bodies during prolonged fasting and starvation. Next, we have the biomedical importance of ketone bodies. There are three important compounds in the body, which are collectively called ketone bodies or acetone bodies or incorrect ketones. These are acetoacetate. It is the primary Kb. Acetone and beta-hydroxybutyrate, it is the second Kb. Ketogenesis starts from mitochondrial acetyl-CoA in liver. Synthesis is mainly hepatic in nature, but liver, due to absence of enzyme thioprase or acetoacetate CoA transferase is unable to utilize Kb as source of energy. We have the 6 properties of ketone bodies. First, they are water-soluble substance. Second, they are synthesized at a relatively low rate in well-nourished individuals. Third, we have the plasma level of ketone bodies. Fourth, we have the urinary level of ketones. Fifth, we have the acetone is synthesized in smaller amounts than other ketone bodies. It is a non-metabolized end product. It is a volatile substance that is excreted by the lungs in the inspired air. Sixth, we have the acetoacetate and beta-hydroxybutyrate are relatively strong acids. Let's proceed to the ketolysis. Ketolysis, it is the utilization or oxidation of ketone bodies. The site of ketolysis is the mitochondrial matrix of extrahepatic tissues, especially kidney cortex, cardiac and skeletal muscles, which normally use ketone bodies as a source of energy in preference to glucose. The brain can utilize ketone bodies as a source of energy only during prolonged starvation, and they provide about 75% of its energy needs. Ketolysis does not occur in first RBCs due to absence of mitochondria. Second, liver, due to absence of thioprase enzyme that is required for the activities of ketone bodies. The ketoacidosis or ketosis is a disorder of excessive production of ketone bodies. Metabolic disorders characterized by a triad of ketonemia, increased ketone bodies in blood, ketonuria, increased ketone bodies in urine, acetone, fruity, odor of breath. There is also dehydration, acidosis, coma, and death if untreated. The ketonemia or increased ketone bodies in blood. In ketosis, the plasma concentration of ketone bodies is well above normal limits. The condition is called ketonemia. The ketonuria or increased ketone bodies in urine. When the concentration of ketone bodies significantly increase above 50 mg in plasma, they appear in urine. The condition is called ketonuria. We have the three acetone or fruity odor of breath. The acetone is also excreted by the lungs and produces a characteristic odor in breath or acetone odor of breath. We have the fourth, metabolic acidosis. A metabolic acidosis is caused by the excessive accumulation of beta-hydroxybutyrate and acetoacetate. Fifth, we have the hyperkalemia. The hyperkalemia acidosis results in the shift of potassium from intracellular to extracellular compartment. Causes of ketosis: prolonged starvation, severe dieting, uncontrolled diabetes mellitus. Diagnosis of ketosis: Rotheras test, estimation of serum electrolytes, acid-base parameters, glucose and urea estimation. Consequences of ketosis: metabolic acidosis, smell of acetone, osmotic diuresis, loss of conscience, dehydration, and coma. That's all. Thank you.